Okay, everyone, this is the second half of the video lecture for Chapter 14 on genetics. If you recall, at the end of the last chapter, we had talked about genotypic versus phenotypic ratios. Oops, sorry about that. And that's what this figure that you see here was. Also, we talked, about Gregor Mendel's early experiments with his pea plants, where he looked at, for example, flower color, plant height, pod shape, seed color, seed texture, all those different things. Now, what we're going to look at next is basically how he developed his second genetic law. Now, remember, his first law was called the law of segregation. And basically, it said that two alleles for a gene located on homologous chromosomes during meiosis separate away from each other or segregate away from each other and end up in separate gametes, sperm or egg. That's what law of segregation says. And he figured that out by looking at results of his crosses for his pea plants. All right. The next law that we're going to talk about is called the law of independent assortment. Okay, law of independent assortment. I'll explain more about what that law actually is here in just a moment. But basically, he derived this law by looking at two separate characters simultaneously in a cross. Okay, let's go look back at his first experiments that he did. For example, when he looked at the cross between a white flowered plant and a purple flowered plant, he was only looking at one character. He was looking at flower color, right? And so that cross was called a monohybrid cross, right? Hybrids, because you have two true breeding contrasting individuals, true breeding for white, true breeding for purple, and we now know that those mean, or true breeding means homozygous, either dominant or recessive, and mono, because we were just looking at one character, in that case, flower color. So that was a monohybrid cross, and the F1 offspring produced were called F1 monohybrids. Okay, now, Mendel, he used that, the monohybrid cross, to develop his law of segregation, like I said. This second law, the law of independent assortment, he developed by doing what he called dihybrid crosses, okay? Prefix di, as we know, means two, right? So now in the case of a dihybrid cross, he is crossing two F1 individuals, and he's looking at, two parent individuals, and he's looking at two characters separately. So he's looking at two characters that are independent of each other, but he's looking at them in the same cross, so simultaneously. Now, why is he doing this? Because he wants to answer a question that he had in his mind. He wanted to know whether or not dominant and recessive alleles are transmitted from one generation to the next independently of each other or as a package. In other words, is there some reason to suspect that recessive alleles go together as a package and dominant alleles go together as a package into the next generation? In other words, maybe you don't have all possible combinations of dominants and recessives recombining in the next generation. Maybe there's some reason biologically why only recessives go together in an individual and only dominants go together in an individual. He wanted to figure out which was the case, okay? He wanted to figure out if assortment was dependent, in which case you would only get recessives together, or you'd only get dominants together, or was assortment independent? All right, independent, in which case you get all possible combinations of dominants and recessives in the next generation. To do this, he had to develop what was called a dihybrid cross. All right. Now, this dihybrid cross is a Punnett square, or we use a Punnett square to show the results of it. It's a little more complicated than the monohybrid cross. If you remember, in the monohybrid cross, you had a Punnett square with 4 cells, right? And those 4 cells showed the potential offspring genotypes you would get from two parents where you're looking at one character only. Well, now he's looking at two characters. And so now you're going to have a Punnett square in the dihybrid cross that has 16 cells in it. Okay, so let's take a look at what I mean. All right, this is basically laying out the question he had and the approach he took to answer that question. Okay, so what we have up here are two parents that contrast, right, in two characters. So we have different color, yellow versus green, and notice we have different texture, smooth peas versus wrinkled. So texture and color are the two characters. That means we have two separate genes, and that's what the Y and the R genes are. So let's say that Y is color and R is texture. So smooth, which this one is dominant, two uppercase Ys. It's homozygous dominant for that, for the gene that for texture, and that gives it a smooth texture. It's also homozygous dominant for color, and so yellow is dominant. This one is homozygous recessive for texture, so it's wrinkled, and homozygous recessive for color, so it's green. So green and wrinkled are recessive, smooth and yellow are dominant. Okay. So that's what he starts off with. He's looking at two parents that contrast in two separate characters. They're true breeding for both. Okay, homozygous for both. All right. So when this parent makes gametes, for those two genes, that gamete or those gametes can only be dominant for both, right? Because uppercase Y, this parent only has uppercase Y's dominance to give here. It also only has uppercase R's dominance to give here. Likewise, this parent is recessive for both, so its gametes are recessive for both of those genes. So when an egg like this is fertilized by a sperm like that, you end up with heterozygotes, dominant recessive, and because yellow and smooth are dominant, all of those look just like the dominant parent. All right, that's the F1. Now here's his question. When these F1s either self-pollinate because they're plants or cross-pollinate with other individuals, what are the possible outcomes? Okay, well, here's his hypothesis of dependent assortment. Now, Gregor Mendel had both of these hypotheses worked out beforehand on paper. He knew what the results would be here, basically, if dependent assortment is how it worked. He knew what the results would be here if independent assortment is how it worked. This is the correct one. We know this is how it works based on his results, all right? But let's look at dependent assortment first. So in the case of dependent assortment, he assumed there might be some reason why in the gametes you can only get sperm, let's say, that carry two dominants or sperm that carry two recessives. In other words, you can't have a sperm that's dominant for one and recessive for the other, right? It's either dominant for both, recessive for both. Same would be true for the eggs. That's what dependent assortment means, all right? If that's the case, you would end up with three individuals that phenotypically look like the parent and one that does not. You get, again, a 3-to-1 ratio of phenotypes. Yellow smooth, three of those for one green wrinkled. Okay. Now, what if assortment is independent? Well, then you would get the same type of gametes you did before. Notice this sperm is the same as that sperm. This sperm is the same as that sperm, right? But then you have, that R should be up there, you have two other types of sperm where you have a mixture of dominant and recessive alleles, okay? So 4 types of sperm, and the same thing in the eggs, 4 different types of eggs. 2, this one and this one, the same as what we had in dependent assortment, but 2 additionals where you have a mixture of dominant recessive alleles that you don't have over here. Again, that's independent assortment. It just means that you have all possible combinations of alleles in the gametes. Well, now, because you have 4 different types of sperm and 4 different types of eggs, 4 times 4 gives you 16. You have a Punnett square with 16 cells, all right? Okay. Well, Gregor Mendel did the experiment, and what he found was this. He found that he had 1, 2, 3, 4 different phenotypes possible in the F2 offspring, and they were present in the ratio he predicted, 9 to 3 to 3 to 1. So 9, 3, 3, 1. All right? Now, that ratio, 9, 3, 3, 1, you only find that when both parents are heterozygous for both genes. So this one's heterozygous for Y, uppercase, lowercase, heterozygous for R, dominant, recessive, okay? When both parents are heterozygous for both, that's the only time you get 9, 3, 3, 1. If one of these parents were homozygous dominant for one of the 2 genes, you would not get 9, 3, 3, 1. So let's see how he worked this out. Let me zoom in a little bit here. All right. So first of all, when you're doing one of these, you have to figure out what the gamete genotypes are going to be. That's the genotypes of the sperm and the eggs, okay? And here's how you do that. So if we had, I'm just going to use letters that are simpler to do here with the Apple Pencil. Let's say you had a parent that's heterozygous for those two genes, right, as our parent in this case was. How do you figure out what all possible combinations of those letters are to produce gametes, sperm and egg? Well, it's simple. It's sort of like the FOIL method in algebra, first, outer, inner, last, right? So here you have one combination, so that's an AT gamete. Here you have a second combination, that's an AT. Here you have a third, and there you have a fourth. So those are the 4 gamete genotypes that are possible from that parent. And I'm going to tell you, this is usually what students screw up when they do these dihybrid crosses, okay? First of all, you have to have gametes that have 2 letters in their genotype, because you're looking at 2 separate characters, 2 separate genes, and you will have 4 different possibilities. Okay, you have to figure out all possible gamete genotypes by doing that method right there. That's the step that students usually mess up when they're doing these dihybrid crosses. Okay, now if your other parent is exactly the same as this parent, the other parent would have exact duplicate gamete genotypes. Okay, all right. So then you just put them across the top and the side. It doesn't matter whether the father's or mother's is on the top of the side, doesn't matter. So we have a sperm here that's homozygous dominant for both genes. Let's say it fertilizes that egg. This is what you get. 2 uppercase Y's right there, 2 uppercase R's right there. You just drop the letters in, right? And that's going to be a yellow, smooth individual. All right, same thing here. Now we have a heterozygote for the R gene for texture, but homozygous dominant for color. So again, it's yellow smooth, 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 yellow smooth. All right. Now, there are 9 of those. 1, 2, 3, 4, 5, 6, 7, 8, 9. That's our 9 right there. 9 of those, 9 out of 16. Okay. Now let's look at the green smooth right here. It should be 3 of those, right? Right here. 1, 2, 3. Should be 3 of the yellow wrinkled. 1, 2, 3. Should be 1 of the green wrinkled right there. All right. So that's our 9, 3, 3, 1. So working these Punnett squares and these dihybrid crosses is not that difficult. You've got to get the gamete genotypes correct. You've got to drop them into each cell correctly. You got to write these letters down correctly. And then you've got to be able to interpret what those letters mean, right? That one's going to be a yellow smooth. This one's going to be yellow wrinkled. This one's going to be a green smooth and a green wrinkled. You have to be able to interpret by dominance and recessive what these genotypes, these letter combinations, are going to result in terms of the phenotype. The phenotypes are the physical appearance. Okay, I would suggest you work out some examples of that, some practice problems with that, by changing the parent genotypes around and seeing what you get, okay? All right, so again, Gregor Mendel knew that if assortment was independent, he would get basically 4 different types of offspring in the F2, present in a ratio of 9:3:1. When he did the experiment, that's exactly what he got. And here you see his actual results, 9:3:3:1, roughly. So he knew assortment was independent. That's his law of independent assortment. Okay. Now, the law of independent assortment states, again, that each pair of alleles segregates independently of each other pair of alleles during gamete formation. I talked about independent assortment back in the meiosis chapter in terms of how homologous chromosomes from our parents line up when we carry out gamete formation. Okay, it's based on what Gregor Mendel found here with these Punnett squares. All right, it does only apply to genes on different non-homologous chromosomes, however. Okay, and there are good reasons for that, which we don't need to get into. Okay, all right. So, Gregor Mendel developed the Punnett square to be able to figure out the likelihood or the probability of getting any one of these possible offspring genotypes, right? So yellow smooth is much more likely than green wrinkled, for example, right? Now, what if you had so a Punnett square where you're looking at 2 characters gives you 16 cells. What if you're looking at 3 characters? That one gives you 64 cells. 4 characters, 256 cells. The number of cells in a Punnett square goes up dramatically and very quickly as you start tacking in more genes or adding in more genes. It becomes unwieldy very quickly. Fortunately, though, we can apply some simple laws of probability to figure out the likelihood of getting any particular genotype in the offspring from these crosses without having to do the Punnett square and counting them all out. Okay, so Mendel's laws of segregation and independent assortment reflect the rules of probability. And there are a number of these rules, but we only care about one of them, and it's very simple. We'll get to it in a minute. So, for example, when tossing a coin, the outcome of one toss has no impact on the outcome of the next toss, correct? If you toss a coin once and you get heads, that in no way indicates that you're going to get tails or won't get tails in the second toss, because the second toss is completely independent of the first, all right? In the same way, the alleles of one gene separate or segregate into gametes independently of the alleles of another gene, all right? Each one is a toss of the coin, basically. So we have something called the multiplication rule that will help us figure out the likelihood of getting an offspring with a given genotype out of all the possible genotypes that there are in the offspring population. The multiplication rule says that the probability that 2 or more independent events will occur together is the product of their individual probabilities. Okay, well, what the heck does that mean? All right, here it is. So let's say that you want to know what the probability is of tossing a coin twice and getting tails both times. Well, you know the probability when you toss it once of getting tails is 0.5, 50%. You know the probability of getting tails the second time is also 0.5 or 50%. So what's the probability under the multiplication rule of getting 2 tails in a row? 0.5 times 0.5, 0.5 squared. So 0.5 times 0.5, 0.25. 25% chance of getting of any 2 tosses turning out to be 2 tails in a row. All right. Likewise, let's say you have friends of yours have 3 children and they're all girls. What's the probability of that? 0.5 times 0.5 times 0.5. Very simple. The answer to that, I think, is 1 in 8, if I remember right. So that's how you figure it out. Okay. Now, segregation in a heterozygous plant is like flipping a coin. Each gamete has a 1/2 chance or 50% chance of carrying the dominant allele and a 50% chance of carrying the recessive. So if we take a look at that, we can see this here. All right, so here we have 2 heterozygotes, uppercase, lowercase, all right? And that means that the sperm, let's say of this individual, half of them carry the dominant, half carry the recessive. Remember, sperm and eggs are haploid, only 1 copy of each gene. The eggs, half dominant, half recessive. So what's the probability of getting a homozygous dominant individual or offspring right here? 0.5 times 0.5, 0.25. 1 of 4 is homozygous dominant. All right, what about homozygous recessive? 0.5 times 0.5, 0.25, right there. Okay, 1 of 4. Now, heterozygotes. There's 2 ways to get a heterozygote. You can have the sperm being recessive and the egg dominant, as in the case here, or the egg being recessive and the sperm dominant, as in the case here. All right, so there's 2 ways to be heterozygous, which means you get 2 out of 4 being heterozygous. Okay, that one is 1 in 4. That one is 1 in 4, or 25%. All right, so because there are 2 ways to have it, we end up with 2 or half, 50% of our 4 offspring being heterozygotes. Okay. All right. So the multiplication rule then explains how it is the alleles come back together randomly during fertilization. It's the same thing. All right. Up to this point, what we've talked about is called classical Mendelian genetics. Okay, classical Mendelian genetics. Mendelian provided the basis of modern genetics by doing these very simple experiments and developing his law of segregation and law of independent assortment, as we've talked about. Now, since Gregor Mendel's time, we have found other situations that are a little more complicated compared to what Gregor Mendel dealt with. Mendel got very lucky in picking those 7 characters in pea plants because they all followed the same basic rules. Law of independent assortment, law of segregation. They were the simplest genetic case. In each of those characters, flower color, pod shape, whatever, you had one gene that came in only 2 versions, 2 alleles, and one of them was completely dominant and the other completely recessive. All right. So if a dominant was present with a recessive and a heterozygote, the recessive had 0 effect. That's called complete dominance. Okay. We've since learned there are other situations, and that's what we're going to talk about in the next part, or the last part, rather, of this chapter. So the relationship between the genotype and the phenotype is rarely as simple as Gregor Mendel found it to be in his pea plants, okay? There are a lot of heritable characters that are not determined simply by one gene with just 2 alleles, for example. In some cases, you have multiple genes working together to determine the phenotype. In some cases, you have a gene that has 8, 10, or a dozen different alleles, not just 2. All right. However, even though we have those more complicated situations, the basic principles of segregation and independent assortment still apply. All right. Okay. We're not saying that anything that Gregor Mendel did was invalid. We're saying that now we're taking the simple things that he sort of dealt with and applying them to more complicated situations. And we find that they still work. So inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations. You can have alleles not being completely dominant or recessive. So you could have a heterozygote that has a dominant and a recessive, but the recessive still has some small effect on the phenotype. Okay? We can have a gene that has more than just 2 alleles, as I mentioned. And we can have a gene that produces multiple different phenotypes because maybe it produces a protein that has multiple different roles to play in the biology of the cell. All right? So there's multiple ways that gene can have an effect, in other words, on the phenotype. Let's talk about dominance first. So first of all, we have what's called complete dominance. Okay, complete dominance happens when phenotypes of the heterozygote and the dominant homozygote are identical. That's exactly what Gregor Mendel saw. You go back and look at his cross for the color of the flowers of the plants. A heterozygote and a homozygous dominant, both purple, look exactly the same. Okay. Now, we also have situations that we've learned of since that we refer to as incomplete dominance. Here, the phenotype of the F1 hybrids is somewhere between the phenotypes of the 2 parental varieties. Okay, I'll show you an example of that. We also have something called codominance, where you have 2 dominant alleles that affect the phenotype in separate, distinguishable ways. So you have 2 alleles for a gene that go into a cell or an organism, and one is not dominant and the other recessive. They're both dominant, and they both actually have separate, distinguishable effects on the phenotype. All right, let's look at an example. So here we have a situation of what's called incomplete dominance, okay? So these are 4 o'clock flowers, a species of plant called a 4 o'clock flower. And notice that we have red flowers and white flowers, and here we have 2 alleles indicated R, uppercase R, okay? So you get 2 R alleles, that gives a red flowered plant. You get 2 W alleles, again, notice that they're both capital letters, indicating that they're dominant as well, and you get a white flowered plant. This plant can only produce gametes that carry the W allele. This one only gametes that carry the R. When they come together, you get a heterozygote, okay? And they're both dominant. They both have separate distinguishable effects. How do we know that? Well, if this one produces red pigment and that one produces white pigment, you mix red and white, what do you get? Pink. So we have a protein producing red pigment and a protein here producing white pigment. You mix them together, and it's just like mixing red and white paint. You literally get pink. All right? That's a case of codominance. Okay. Now, look what happens in the F2. If these guys reproduce, half their gametes carry W allele, half carry the R. And so now we can end up with all 3 phenotypes. Red, if these 2 gametes come together. Pink, if these 2 or if these 2 come together. And white, if that one and that one come together. So these are situations that Mendel didn't deal with. Had he dealt with something like this, he might have been able to figure it out. Again, given the lack of information he had at his availability at that time, or available to him, maybe he wouldn't. I don't know, but this is just something that Gregor Mendel didn't have to deal with in his pea plants because they showed complete dominance. All right. Now, we also have situations where we have multiple alleles. So you have a gene, for example, that has maybe 3 alleles or 4 or a dozen, not just 2 like Gregor Mendel dealt with. Here you get a lot more possibilities in terms of the phenotypes you can imagine. Great example of that is the ABO blood group in humans. Okay, so there you have type A blood, type B blood, type AB blood, or type O blood. All right, why do we have 4 different phenotypes as far as blood group goes? It's because there are 3 different alleles that code for this. Okay, so what are blood types? Blood types have to do with the type of carbohydrate that we find on our red blood cells, all right? Or whether we have that carbohydrate at all. So we have all kinds of carbohydrates and proteins on the surfaces of our cells. Our blood cells have basically it's called a glycoprotein. It's a protein with a small carbohydrate sugar on one end of it, and it juts off of the side or off of the cell membrane, the outer cell membrane of our cells, okay, blood cells. Okay, now there's an enzyme, a protein, that codes for whether or not that glycoprotein gets placed there, all right? So we have what are called, well, let me just go ahead and show you the next figure here, okay? So we have a gene, let's call it the I gene, that codes for whether or not that protein or that glycoprotein gets put onto our cells. If you have the IA allele of that, you have the A glycoprotein. They're saying carbohydrate, but it's a carbohydrate attached to a protein, okay? Shaped like that, let's say. If you have the B allele, you get the B version of that glycoprotein. The I allele produces no glycoprotein on the surface of the blood cell, all right? Now, let's look at the different genotypes. If you are IAIA or IAI, in either of those cases, you end up with the A glycoprotein on your blood cells, right? And you have type A blood. That's your phenotype for blood type. All right, if you're IBIB or IBI, you get the B glycoprotein. If you are IAIB, you get both. All right, that is a codominant situation. You get separate distinguishable effects of the 2 separate alleles. You get an A glycoprotein and a B, and so you end up with AB blood. However, if you're homozygous for the I allele, lowercase I, then you end up with type O blood because you don't have glycoproteins on your blood cells, okay? So here we have 3 separate alleles for the same gene that have separate distinguishable effects. So it's a multiple allele situation, but also, as I said, a codominant situation as well. All right. Now, next thing, what about 2 or more genes? So in every case we've talked about up to this point, we've talked about just one gene basically, okay? Now, what if you have 2 or more genes that are sort of working together, working in concert together, okay? And there are some traits that are actually determined by 2 or more genes that we know of today. This can lead to a situation referred to as epistasis, okay? In epistasis, a gene at one locus alters the phenotypic expression of a gene at a second locus. So it's almost like one gene is controlling the expression of another gene, all right? Great example of this is coat color in certain types of mammals, certain species of mammals. For example, Labrador retrievers, okay, and a lot of other mammals, the coat color actually depends on 2 separate genes. One gene determines the pigment color, all right, and we'll have, let's say, an allele, uppercase allele or dominant allele that will code for black, and a recessive that will code for brown. Okay, so let me just write that in real quick. Codes for black, that codes for brown. And so we have the B gene then coding for the color of the pigment. We have another gene, let's call it the E gene, that codes for whether or not the pigment gets deposited into the fur. So we have a dominant and a recessive for a second gene, the E gene, and the dominant codes for pigment being present in the fur, and the recessive codes for it being absent. Okay, you might imagine the E gene here can affect the expression of the B gene, okay, as we'll see. Let's take a look. All right, so we have two parents that are heterozygous for both the B and the E genes, right? Dominant recessive, dominant recessive. Okay, and because black is dominant and E, or the dominant gene for, the dominant allele for the E gene codes for the pigment being present in the fur, we see the fur being colored black because black is dominant, and E says that the dominant pigment, or the pigment rather, goes into the fur. All right, that's the case with both of these parent dogs here. Okay, so look at the possible gametes from those two parents. Remember, all possible gamete genotypes there. This sperm fertilizes that egg. We get a dog that's dominant for both. So the two uppercase B's means that the pigment is black. The two uppercase E's means that the pigment actually is deposited into the fur, so you see it. All right, we get a black dog as a result. That same thing is true for all of these black dogs here, right? Uppercase B, uppercase E in every single one of them. Now look at this dog, brown dog. Why is it brown? It's brown because for two reasons. First of all, we have pigment going into the fur. That's what the two dominant E's tell you. And that pigment is brown because of the two recessive B's. Same thing here. E says it goes in the fur. Lowercase b say it's brown. Same thing there. What about these four dogs? Why are they white, or whatever they call it? Well, the color of the pigment actually doesn't matter here. In every case, we have homozygous recessive for the E gene, which means that pigment doesn't go into the fur. If pigment did go into the fur in this dog, it would be black. Also black there, also black there. If pigment went in the fur in this dog, it would be brown. All right, so you see how the E gene, which codes for whether or not pigment goes into the fur, is affecting the expression of the B gene, which codes for the color of the pigment, right? The E gene is sort of controlling the B gene. That's epistasis. Mendel did not deal with that with the characters he looked at in pea plants. Okay. All right. This brings us to something called polygenic inheritance. In polygenic inheritance, you have a lot of variation possible because you have multiple genes, polygenic, involved. Okay. This also brings us to what we call quantitative characters. So quantitative characters are those that vary, in terms of their phenotype, vary along a continuum. You don't just have, for example, purple flowered plants and white flowered plants, okay? Maybe you have purple and then all shades possible between that and white. So what you end up with then is basically a normal curve, okay, a bell curve. So you have, down here on the x-axis, you have some range of phenotypic characters, color, height, stature, whatever. And instead of just having two types, you have what's called a frequency distribution, where you have the average value located there in the middle. And most individuals, located somewhere near that, have something near that average value. But there are some individuals that have an extreme value here, some here. If this is height, let's say, then you have some really short individuals, a few of them, a few very tall individuals, but most individuals are somewhere in the middle around the average, okay? Average height. That is what you get with polygenic inheritance. That is quantitative variation along some continuum, okay? All right, skin color in humans is actually a very good example of this. Now, here I'm talking about skin color or skin tone within a racial group, say within Caucasians or within Asians or within Africans, for example. All right, not across racial boundaries, that's different. Okay, we know that within a racial group, there are three separate genes, let's just call them A, B, and C, that code for skin tone. All right, and this is something that's referred to as additive because the more dominance you have, the darker the skin tone, all right? So here we have three dominants, gives you darker skin tone. Three recessives gives you lighter So we have that basic skin tone. The shading of the block tells you what the skin tone is. Two parents are being heterozygous for all three genes have the same skin tone. But look at all the different types of offspring they can have in terms of skin tone. All right. So here we have a 64 cell Punnett square, again, because now we're looking at three genes, like I said before, right? And let's say that sperm fertilizes that egg, you get an individual that's the lightest skin tone possible. Let's say that sperm, I can get to work. That sperm fertilizes that egg, you get the darkest skin tone possible. That's this extreme and this extreme. But everybody else falls out somewhere near the middle. This is our average. 20 of 64 have that skin tone. 15 of 64 respectively have these two. 6 of 64 have those, right? So most individuals are somewhere here. And so what we end up with then is a curve, right? Based on these frequencies, like I showed you before. So this is a good example of how you can have two parents that have very similar skin tone. They can have a child that has very light skin tone and one that has very dark, or something that's somewhere in between. Okay, they can run the gamut. My point is, with quantitative variation with multiple genes involved, you get a lot more variation possible than in the simple cases like what Mendel dealt with his pea plants. One gene, only two forms or versions, one completely dominant the other. You don't get a lot of variation. You get either or variation, either purple or white. Okay. Last thing to talk about in this chapter is the question of nature versus nurture. So everybody's heard this expression, nature versus nurture. It has to do with the extent to which we think genetics or the environment affect how we turn out. Well, there are a lot of people that have argued that the way we are is hardwired into our genes. It's all genetic. It's all nature. Others say, but it also depends, how we turn out also depends on the environment we grow up in, right? The kind of parents we have, that sort of thing. Our upbringing, that would be the nurture part of it. Well, it turns out in most cases it's probably a combination of the two, okay? So another departure from Mendelian genetics arises when the phenotype for a character depends on the environment as well as the genotype. So genotype is genetic makeup, phenotype is physical appearance. We now know that there are a lot of cases where the environment can modify the phenotype to something above and beyond what the genotype is sort of dictating it to be. The phenotypic range, how much variation you get in the phenotype, is broadest for polygenic characters, those that are coded for by multiple genes, like skin tone, for example. Okay? Traits that depend on multiple genes combined with environmental influences are called multifactorial. Okay? So characters or traits that are a result of the combination of multiple genes and environmental influences are called multifactorial. All right, and we get a lot of variation possible here. Okay, so good example of that. I forget what kind of plant this is. It's down here in the southeast. I can't remember. Anyway, if you look at these two plants, obviously you see that their flower color is different, pink versus blue. All right, but they have the same genotype for flower color. So because they have exactly the same genotype for flower color and their flower colors are different, there must be something else causing the flower colors to be different, something other than the genotype. It turns out it's soil pH. And I forget which one's which, but maybe acidic soils give you pink flowers and more basic soils give you blue flowers, or it could be the other way around. I don't remember. But the point is that you have two plants that for flower color are identical in terms of their genotype. They have the same alleles for that. But you put them in different environments, different soil pHs, and the environment modifies the phenotype, okay? This is an example of a multifactorial trait, okay, or character, and there are a lot of things like that. Think about skin tone. Think about skin tone in Caucasians. In January, I had my lightest skin tone because I've been cooped up in the house all winter long, but by July and August, I've been to the beach for a few times, or a few visits. I've gotten some tan, and now my skin tone is a little bit darker. Genetically, my skin tone is what it is, but the environment, how much exposure I had to sunlight, can affect that and modify that. That's an example of a multifactorial character, okay? So last thing here, An organism's phenotype includes its physical appearance, its internal anatomy, its physiology, its behavior, possibly even to a certain extent intelligence. All of these things are aspects of an organism's phenotype. The phenotype then reflects its overall genotype and its unique environmental history. Okay, what kind of environment it's grown up in, all sorts of environmental influences can affect the phenotype. So when you look at the phenotype of an organism, it's not just due to the genetics or the genotype, it's also a reflection of the environmental influences on that organism during its development as well. Okay? All right, that is the end of chapter 14.